My laboratory is at MGH and the Broad Institute, and we study epigenetics. We've become increasingly interested in the past decade in how epigenetic lesions lead to cancer. Here's my disclosure form. So I want to talk about a couple things today. First, I want to spend a little time giving you some background into chromatin biology and epigenetics. And then I want to tell two stories. One about how epigenetic mechanisms can lead to initiation of brain tumors. And the second is how epigenetic plasticity can lead to evolution, adaptation, and ultimately drug resistance in brain tumors. I think they fit together well and sort of exemplify what's going on in epigenetic mechanisms in cancer. So a succession of developments over the past decade have really highlighted sort of the centrality of epigenetics to cancer biology. We now realize that chromatin regulators are among the most frequently inactivated genes in the genome in cancer. Right? These have been highlighted by the Cancer Genome Atlas studies and all these other studies of exomes and mutations in cancer. We know that gene activity and methylation is globally altered in cancer. We know that there's extensive phenotypic heterogeneity among different cells within a tumor. And finally, we're starting to get indications that epigenetic drugs are looking very promising in cancer and in some cases have been approved. That said, these points I make up here fall short of clarifying the mechanisms by which epigenetic alterations lead to cancer. They don't really explain to us how this process is happening. They don't allow us to intervene in a rational way. So I think that the central, probably the most important goal in our field of cancer epigenetics right now is really to take this further and understand the underlying mechanisms at play so we can attack them in sort of a targeted and rational way. And that's the focus of a lot of what I want to talk about. But before, I want to step back and just say, what is epigenetics? What are these chromatin factors doing that are so frequently mutated in cancer? What is going on here? I think I would start at the level of the human genome. The human genome is enormous, right? It's 3 billion bases that stretch about 6 feet, linear DNA. Needs to be compacted into a nucleus that's the size of the sharp end of a pin. Sure, the genome has genes, has about 20,000 genes, protein coding genes, but those actually account for only 1% of the sequence. The other 99% has sort of remained kind of mysterious. Even the Human Genome Project did not clarify what was going on there. People thought it was junk, but it's become very recently clear that this other 99% is dispersed with regulatory elements, enhancers, switches, and other elements that control gene activity. Remember, outside of cancer, in your normal development, this one genome is present in every cell type, and they all have the same genome sequence. So perhaps it becomes almost expected that there would be enormous regulatory mechanisms that control precisely which genes are on and off in which cell type. And this is what the genome is made up of. These regulatory elements are activated by developmental transcription factors or other factors that just switch them and switch the genes precisely in very much the right context. So what is chromatin doing? Remember, this is a 6-foot-long string of DNA. So chromatin is packaging all of this genome. There are these, of course, these spools that are sort of shown here. The nucleosomes around which the DNA is wrapped. Chromatin can exist in different states. There are sort of active states where the chromatin is wrapped a little more loosely, and you might have an active promoter bound by transcription factors and polymerase. You might have enhancers that are active and accessible. Alternatively, chromatin can exist in this very compacted state that sort of organizes very tightly all of these nucleosome spools and becomes quite inaccessible to the regulatory factors that would like to turn on a particular site. But we draw here an insulator, which I'll come back to, but the genome is also sort of dispersed with insulator elements that sort of demarcate, allow this region over here to be distinctly regulated from this region over here, right? So this region here can be compacted and inactive, and this region can be made accessible. So what's going on, at the molecular level? I'm not going to spend a lot of time on this, but the field has identified a sort of an enormous number of regulatory enzymes, HDACs, histone acetylases that add or remove modifications on the histone tails on the DNA. And these on states have particular character, and there's certain enzymes that sort of promote the on states. These off states have very distinct regulators and distinct structure. And all of this, importantly, exists in some dynamic equilibrium. So this is a key point I want to make, is that in different cell types, different regions are open or closed. And even in the same cell type, particularly early in development, we need to have the permissivity in our chromatin structure to switch between states as cells develop, alternate their states, differentiate, or respond to some environmental cue. So this is critical. So homeostatic chromatin, the normal state of chromatin, permits the appropriate transitions in gene activity in global programs. Okay? And I draw a very simple schematic. Imagine this little ball is a cell. These are sort of energy barriers to the cell, right? So this cell can switch into a state. Here, maybe it turns on a gene, or maybe it differentiates a little bit, but it cannot switch into these other states of other cell types, right? Because chromatin acts as a barrier, allowing just the right transitions, right? And you can imagine this at one gene, where there's a gene that is, its promoter is regulated by repressors like EZH2 and K27 trimethylation, and activators like MLL and K4 methylation, right? So these kind of sit in a balanced or poised state that can allow just the right amount of permissiveness, okay? This is what I think the field is, what's emerging out of the field of cancer epigenetics is that a couple things very frequently happen. And when you think about these chromatin regulator mutations, you might think about it in this context. You can extend the same single gene model to a network, and it can allow a network of growth signaling, growth regulation to switch on or off. And again, chromatin is just the right barrier between alternating these states. It helps, it sort of acts as a resistor that helps stabilize these states, but allows the right transitions. Okay, and what I was just saying is that in cancer, this homeostasis is, I would argue, almost universally disrupted. And I would propose to you that many of the mutations and other events that you're seeing, or environmental effects, are disrupting this homeostasis. And I'll give you a couple examples. So one way this homeostasis might be disrupted is that chromatin switches into a restrictive state. You can imagine that this barrier could be amplified, thus keeping these cells here. This gene switches from this poised, balanced state to now a very stably silent state. And this is, I think a decent example of this is the gain-of-function EZH2 mutations that one sees in lymphoma. Okay, and I could give you other examples if you wanted to go through it, but I'm just trying to make this straightforward. Right? Gene silenced, it can't come on, cells can't switch states, restrictive chromatin. I think you can find a lot of examples where restrictive, with some of these mutations that you see in chromatin regulators, and I would argue by extension, environmental effects that alter chromatin state would act in this way. Okay, but there's more. Alternatively, one could imagine that you could have alterations, either genetic mutations or environmental events that make the chromatin too permissive. Now these barriers are lost, and this cell can turn on genes that it shouldn't turn on. It turns on them at the wrong time. It's spurious. It's permissive. An example might be a loss of function of this repressor EZH2. Now you get spurious gene activity. Okay, so leading to a plastic state. This is really what I want to focus on today, because our work in glioblastoma, I think, is providing some very strong examples of how plasticity, such as this, can be driving tumorigenesis. I want to frame this and sort of, draw some parallels to genetics, and also just sort of, make the argument that cancer epigenetics is a big piece of the puzzle, right? A lot of us think about cancer as a genetic model of cancer, a genetic disease, right? Wherein mutations to the DNA turn genes on or they turn genes off, right? But look, we're learning in epigenetics that these modifications to the DNA, alterations to the chromatin structure, chromatin folder, can also turn genes on or off, okay? And I'll take it a little further. In the plasticity case, I think it's very familiar, wouldn't take much convincing, that there are certain mutagens or mutatable, hypermutatable states where cells acquire a lot of mutations, right? A lot of these mutations are passengers. They don't really contribute to the tumor, but some of them will be drivers. And I'm going to make the argument today that there's exactly the same thing going on in cancer epigenetics, that there are certain triggers that increase epigenetic plasticity. They allow a lot of epigenetic switches to be thrown in aberrant stochastic ways, A lot of those events will be passengers. They will be inconsequential and they will not contribute to the fitness of a tumor. But some of those events will be drivers. By triggers, I mean genetic changes, such as mutations to chromatin regulators, but also environmental changes, stress, or metabolic changes that alter some of the activities of the enzymes, right? So these stressors, I would say, can, in general, induce plasticity in the epigenetic machinery. Okay, and now I'm going to go on and try to give you some pretty specific examples of what I'm talking about. So in the first, I want to talk about how IDH mutations in glioma lead to a loss of oncogene insulation, thus driving tumor initiation. Okay? Gliomas are malignant brain tumors. Glioblastomas in themselves are the most frequent. They're the cause of about 13,000 deaths per year. So despite radiation and chemotherapy, median survival is only 15 months. The tumors are resected, yet they universally will relapse at different places in the brain, telling you that by the time you've gotten to this point, there are infiltrating cells probably throughout a lot of the brain. This helps us understand why this is such a refractory and incurable tumor. Now, there's a lot of different kinds of brain tumors, a lot of different kinds of gliomas even. So this schematic just describes some of the different classes. I start with a progenitor cell. We don't really know which one this is. It might be different in different cases, but one lesion is the IDH lesion. So this is an IDH mutation. I'll talk about this a little bit. And this gives rise to this entire class of lower grade gliomas. They have a better prognosis, yet they also remain incurable. Okay? So oligodendroglioma and diffuse astrocytoma. So for the first part of my talk with tumor initiation, I'm going to talk about IDH, how IDH initiates this class of tumor. For the second part of my talk, I'm going to focus on glioblastoma, which has some of these more canonical lesions like amplification of RTKs or other kind of genetic activation of RTKs. I'm going to talk about glioblastoma in part 2. I'm not going to talk about pediatric DIPGs and pediatric glioblastomas, but suffice to say, it helps illustrate why brain tumors are such a powerful model for studying epigenetics, because at each turn you see different deregulation epigenetic pathways, whether it's IDH or in this case, histone mutations and so on. This is why this is a very compelling model. I'm going to talk about these two pieces here. So first, IDH mutations. So IDH is a metabolic enzyme. So it's a bit of a surprise. I'm sure many of you remember when the TCGA and actually also sequencing work in the Vogelstein group at Hopkins turned up IDH as one of the most frequently mutated genes in brain tumors. So, bit of a surprise, but pretty soon it was learned that the IDH mutations are not inactivating mutations. Rather, they're recurrent gain-of-function mutations that give this IDH enzyme an entirely new function. And IDH is turned later to not only be mutated in glioma, but also in leukemia, sarcomas, cholangiocarcinoma, and other cases too. Now, the mutant IDH now generates a neo-oncometabolite, this new small molecule 2-HG, 2-hydroxyglutarate. 2-HG is a potent inhibitor of large classes of enzymes, including demethylases. For the purposes today, I'm going to talk about the effect of mutant IDH, its production of 2-HG on DNA demethylases. When you have an IDH mutation, which is probably about the earliest mutation that occurs in some progenitor cell in this disease, a very early initiating event. First thing that happens is you can no longer demethylate your DNA. So you can't properly maintain the homeostatic levels of DNA methylation. And over time, I don't have proof for this. We don't really know, but I suspect this is a long and slow process, maybe many years. We could talk about that. But what's clear is that over time, your DNA gets progressively hypermethylated. So all IDH mutant tumors have this hypermethylated state. There's just too many methyl tags on the DNA. So for a long time, there's a lot of tumors that have hypermethylation. It's kind of been thought that methylation, it must be silencing genes by acting at gene promoters and silencing genes. There's some examples of that, P16, but But there's not a lot of examples of that. So it doesn't seem that this idea that methylation is silencing genes is sufficient to really explain how it could be driving cancer. So we kind of thought about another mechanism, and that's what I want to talk to you about today. So CTCF is a very interesting DNA binding protein that acts as an insulator in our genome, and I'm going to tell you a lot about it. But the first thing I want to tell you about is that CTCF recognizes this very specific DNA motif shown here. And this motif can have a couple of CPGs in it. And if either of those the Cs are methylated, the cytosine methylation, CTCF can no longer bind to its site. Okay, so methylation will knock this factor off. So we took a bunch of gliomas from IDH wild type and IDH mutant gliomas. We collaborate with the surgeons and pathologists at MGH. And we used ChIP-seq, the simple pull-down method, and sequencing to map where is CTCF binding in the human genome in these different tumors. And this is just, these scans just tell you where the sites are. So here's a site in the genome, another site in the genome, another site in the genome, where in these different tumors, here's a bunch of wild-type tumors, you can see nice binding of CTCF, okay? Perhaps you can see that in the IDH mutant tumors, this CTCF site right here is completely lost. And in fact, we look sort of globally, and we can find that there's about 600 different CTCF sites in the genome that seem to recurrently lose their binding in IDH mutant gliomas, okay? And these sites are hypermethylated. So this is sort of setting up a model that another thing that DNA methylation is doing is it's methylating a bunch of CTCF sites and it's displacing CTCF from its binding, okay? So why would we care? CTCF is a really important protein. It does one of the most fundamental jobs in our genome regulation. You remember I told you about all those enhancers that I said that these switches, there's about 1 million of them regulating these 20,000 genes? You might ask yourself, how do these enhancers get targeted to the right gene, and how do they not go to the wrong place or the wrong gene, and how is that controlled? It turns out that our genome is folded up into a series of loops, chromosomal loops. Some people call them chromosomal domains. Their size is about 100 kilobases to maybe a megabase in size. And these loops are anchored by CTCF dimers. So if you will, CTCF grabs onto a piece of DNA here, and it grabs onto a piece of DNA here, and there's a loop here, and CTCF comes together and it dimerizes. There's been a lot of debate of exactly how this happens. There might be extrusion and so on, but at the end of the day, you've got CTCF here and a little shoestring loop sitting up here. And there are tens of thousands of loops in our genome. This right here, this purple thing, is supposed to be an enhancer. It's a switch, turns on a gene, right? There's a lot of them, right? They come on and off in different cell types, but there needs to be a mechanism by which they regulate the right gene. So this enhancer in this loop can turn on this gene here, this green gene, because it's in the same loop and sort of structurally it's able to access it. But what we're learning is that this enhancer can't touch this neighboring gene because it's in a different loop. Even if it's relatively close to this neighboring gene, if CTCF is in between and it's separated into two separate loops, the enhancer can't activate that gene, right? So in a way, this sort of enforces fidelity in enhancer activity and allows enhancers just to regulate the right gene. And you might think of these loops as sort of the functional unit of the human genome, right? These loops of 100 kb to maybe a megabase, okay? Remember I told you, though, that in glioma we're losing the CTCF sites, right? So these CTCF sites, a lot of them, hundreds of them, are getting methylated now, and CTCF's falling off. So this suggests that you're going to start losing some of these loops. If you lose a CTCF site in between two loops, you could predict that now these two separate loops will refold into one big loop. And this will allow promiscuity of enhancers to now begin to maybe regulate the wrong genes. And I don't want to go into all the details, but we actually analyzed a very large set of RNA-seq data from GTEx for normal brain and from TCGA for brain tumors. And there's a clear signature. If you look at normal brain, you can see you can see correlations between genes, and they get co-regulated. But when they're in a different loop, when they're separated by a CTCF, you break that correlation. You separate them out, right? The gene on the other side of the loop is regulated differently from the gene in the neighboring loop. But if you look at the brain tumors, we see a global signature that there seems to be a deregulation of this process, and not all genes, but some genes that lie in neighboring loops that should be insulated from each other start correlating with each other. It's basically just a signature that tells us there's a global disruption of this structure and this functional demarcation of units, a global disruption in IDH mutant gliomas. Okay. But what I want to show you is one of the genes, when we ranked the genes that were most disrupted, one gene came out at the very top and it grabbed our attention. That gene was PDGFRA. And PDGFRA, of course, is a canonical oncogene in brain tumors and other cancers as well. In glioblastomas that are IDH wild type, PDGFRA is often copy number amplified. You get lots of copies of the locus, and as you can imagine, this leads to very high expression of PDGFRA. But in these IDH mutant lower grade gliomas, PDGFRA is never amplified. It's always got its normal copy number. The locus is genetically completely normal. Okay, but I'm going to show you that it's being deregulated and turned on epigenetically. Okay. So this picture here, excuse, it's a little complicated, but basically this is a piece of genomic region of about 1.5 kilobases of DNA. Here is the PDGFRA gene right here. And there's a couple other genes. This signal right here, this is high C data. This is data that basically tells you about whether pieces of DNA are close to each other in space inside the nucleus or far away. Are they able to contact each other? And the answer is that the DNA over here can kind of contact each other, and the DNA over here can contact itself. But there's actually, we found very little interaction across these two regions. And it turns out there's a CTCF site there. In a sense, this CTCF site, based on high C sort of mapping data, is one of those insulators. It's one of those anchors of the loops. Okay? Now, why does that matter? Well, it turns out that there's a very large enhancer, a super enhancer, if you will, that's about a megabase away from PDGFRA. It's turning on FIP1L1 and some other genes, this very potent enhancer. And it's a neural enhancer. It's on in it's on in different cell types, but it's on in neural lineages very generally. And it's a big, potent on switch. But it doesn't matter because PDGFRA is separated, remember, by the CTCF site. It's a separate loop. This guy can't touch PDGFRA. But we wondered, is it likely, is it possible that we're losing CTCF and we're losing this loop structure in the gliomas, in the IDH mutant gliomas? And that's what I want to show you. So this data I just showed you, if you didn't like it, I'll show it to you in another way. Here's PDGFRA, oncogene, critical growth-driving gene. Like many critical growth-driving genes, it's sort of isolated very carefully in a chromosomal loop, like this. And it's separated from that enhancer I told you about. That enhancer is about a megabase away. It can't touch PDGFRA. And in fact, I can show you data that if you look at wild-type gliomas, this CTCF site right here is totally unmethylated. There's no methylation of the DNA. And I can tell you that CTCF is bound there just fine. But now if you look at IDH mutant gliomas, all of a sudden the methylation is very high. So this exact CTCF site, that exact CPG in the motif is getting methylated. Okay? CTCF binding is being reduced. And I think this is probably the most important result here is a 3C interaction data. This plot here basically tells you whether two loci can physically contact each other within the cell's nucleus. We use a cross-linking, restriction, and ligation to basically ask about physical proximity. And I don't want to go into all the details, but look, in the wild-type cells, this enhancer never contacts PDGFRA. I've been telling you that for a while, because CTCF has insulated them in separate loops. But in the mutant cells, we see a very strong physical interaction between these two. It's basically telling you you're losing this insulation between loops. And now we have to redraw the locus in the IDH mutant tumors. It's refolded, right? These two loops of your shoestring have merged into one. The CTCF site is methylated, as I told you, and now the enhancer is able to contact PDGFRA promoter, and we think it turns on this gene, and I'll give you some evidence for that, okay? Indeed, in IDH mutant gliomas, PDGFRA expression is very high. In fact, it's comparable in expression to those glioblastomas with copy number amplifications of PDGFRA, yet its locus is wild type. And I think this helps explain why. Okay, so let me give you some more evidence for this. So one of the issues is if you take, you might hypothesize that, well, if you could remove the DNA methylation with a DNA demethylating agent like Vidasa or 5-azacitidine used for AML, can you restore this loop, right? You could hit this, you could unmethylate, CTCF could come back in, and in fact we find exactly this. When we treat, we work with glioma spheres, with glioma stem cells that we culture in the laboratory. If we treat those cells, it's an in vitro experiment albeit, we treat those cells, we can see that the CTCF comes back and PDGFRA expression goes down through the floor, okay? So the demethylating agent seems to restore a native conformation, at least in these models that we work on in the laboratory, precisely as expected. But there's another experiment you'd probably like to see, and that is, what if we took a wild type tumor that has this normal loop structure, right? So here's we've taken glioblastomas or gliomas. We have the spheres. We grow these spherogenic cultures in the laboratory. When we take CRISPR-Cas9, and now we mutate the CTCF site. Remember everything I've told you before, we're methylating the CTCF site to inactivate it. But this suggests that if you were just to excise the CTCF site, you'd have an effect. So we did this experiment. And in fact, what we see is that PDGFRA, well, we see evidence that there's a refolding, and I'll show you some of the data. So first, when we mutate this site, the first thing we see is that you lose CTCF binding, exactly as you expect. You mutate, you cut a couple bases out of the CTCF motif, CTCF binding is lost. The second piece of data to show you is that PDGFRA expression is doubled. The protein level expression gets doubled. And this is pretty striking. I would point out that this CTCF site is about 200 kilobases away from the PDGFRA gene. And all we did was we changed about 2 or 3 bases in that CTCF binding site, and PDGFRA expression gets doubled. More so, the cells actually start growing faster. And in fact, our glioma sphere model, the cells now start growing twice as fast as the control. And this increase in growth, it seems to be PDGFRA expression dependent, because if we add a PDGFRA inhibitor, dasatinib, you go back to baseline. Okay? So I think this helps illustrate, how a switch in the loop structure that allows this enhancer to turn on PDGFRA turns on the gene and actually leads to a growth advantage in a cell. All the last work was by Will Flavahan and Yotam Dreyer. It's been recently published in Nature, and you can read about that. We also, I'll share some unpublished data that we recently collected in collaboration with George Dimitri and Jason Hornick in gastrointestinal stromal tumor that I think helps suggest this may be a general mechanism. So GIST, I think many of you are familiar with, have this sort of KIT gain of function mutation. About 3/4 of GISTs have this genetic alteration to KIT, to switch KIT on. But there's about 15%, it's relatively rare, but there is a subset of GISTs that have an SDH mutation. It's not IDH, it's SDH. It makes succinate. It doesn't make 2-HG, but it makes succinate. But succinate is also an inhibitor of the DNA demethylases. And these tumors are also hypermethylated, okay? So we took a look at the locus, and KIT not particularly constrained in a loop. But it turns out there's an enhancer nearby that is very tightly constrained in a loop in GIST cell lines, okay, or in GIST tumors. What we found, though, is that if we took a series of GIST tumors that were SDH wild type, they've got normal methylation, nice binding of CTCF here, and it's unmethylated. But if we then compared those to a series of GIST tumors that were the hypermethylated type, the SDH mutant type, all of a sudden we see that this binding site gets methylated and CTCF falls off, suggesting that here again we have an example of sort of an epigenetic refolding of a locus. In this case, you've untethered an enhancer and you've allowed it to come turn on KIT. And in fact, if you look at GIST, that are KIT mutant, they have, of course, very high KIT expression. But the SDH mutant GISTs also have very high KIT activity, even though they don't have any lesions of the KIT locus itself. You've epigenetically activated, okay? All right, so we think this is a new cancer-causing mechanism. It's an epigenetic way to activate oncogenes. In glioma, right, I've told you in high-grade glioblastomas, often you amplify the PDGFRA locus as a genetic mechanism. But in lower-grade gliomas, you have this IDH mutation that drops out an insulator and epigenetically switches on PDGFRA. We've got a very parallel example in GIST. We're studying other tumors as well, as you can imagine, to see if similar mechanisms might be at play there too. And I think it's worth making the quick point that some of the precision medicine diagnostics, we think about looking for PDGFRA amplification in different tumors or mutations to oncogene that might switch them on. But I think some of this work suggests that there may be epigenetic ways that stably and very strikingly upregulate oncogenes, and we need to consider that from a therapeutic perspective. These may be indications to use some of these targeted therapies. I want to go back to some of this plasticity I talked to you about earlier, okay, sort of the mechanism. How does this all happen? So some of the questions you might be thinking about now is insulator disruption targeted right to the PDGFRA locus? I think the answer is no. I think it's probably pretty random. I don't think that it's such a smart mechanism. There's 20,000 CTCF anchored loops in the genome, and I told you there's about 600 sites lost in glioma. So I think this is probably a pretty stochastic process where different loops get dropped off in different cells at different times. What about stability? You know, geneticists are in good shape here because a genetic mutation is mitotically passed on. It's heritable, right? Mitotically heritable. What about an epigenetic lesion? Well, there's a lot of debate in the field, but I think this one's pretty good because the switch between CTCF binding and methylation underlies parent of origin imprinting. It's why you have two different alleles from your maternal or your paternal, that one gene's expressed from one allele and one's not. This is due to one has CTCF and the other has methylation. So this is really stable states. So the switch that I show you when you switch an insulator, you knock out an insulator and you methylate it, should be heritable mitotically, right? Through cells, through many generations, perhaps in cancer initiation. So this leads us to the following model for sort of stochastic insulator disruption. And you may find it looks a little similar to some of the genetic models that you think about in genetic mutations, right? So we think the very early event, IDH mutation, I could give you evidence from Joe Costello and other work. This happens, maybe the first thing that happens when, you know, way before you guys, way before you diagnose a tumor. Leads to hypermethylation in some OP progenitor cell of some type. It's probably slow cycling progenitor cell. There will be different insulators lost because the methylation might hit this CTCF and might hit that CTCF. In probably the majority of cases, that will be inconsequential. You'll lose an insulator, but there's no enhancer, there's no gene, or maybe there's a gene, but the gene's not an oncogene. It will be like a passenger, right? Inconsequential. But at some point in some cell in this lineage of slowly dividing lineage, I think you're going to lose the PDGFRA insulator. And this is going to be selected for. This is going to be a driver event. The cells are going to gain a proliferation advantage. They're going to expand the population and ultimately acquire the two more mutations that are needed to cause the lower grade glioma. So in a sense, very parallel to genetic mutations, but epigenetic mutations. And it makes one other point, and that is that it's kind of a hit and run mechanism if you think about it. Once you've methylated this PDGFRA locus, an IDH inhibitor is not really going to help you, right? Because you've already made the switch. So I think the tumors are no longer, at least this doesn't count for everything, but in the gliomas, we suspect the tumors are no longer going to be IDH dependent. So I think we'd have to think about other modalities to sort of switch them back. Okay, so for the last part of my talk now, I want to switch over and talk about glioblastoma. And I want to talk about how another form of plasticity, epigenetic plasticity, can lead to drug resistance. And this is work, well, actually, well, I'm going to start out by, this is work by a number of people in the lab, kind of started out here. We know from a number of colleagues in the field, Fine, Dirks, Rich lab, that the glioblastomas are propagated by stem-like cells that are very invasive and they're refractory to therapy. Okay? So one of the missions of my laboratory the last quite a few years now has been to sort of define the circuits. We wanted to understand the transcription factors, the chromatin factors, the elements that are sustaining the GSCs, if you will, the glioma stem cells. What sustains the regulatory circuits? This is work of Esther Reinbe, Mario Suva, and Anup Patel in the lab over the years, have really defined like a core circuitry that underlies these cells. But more recently, Brian Lau was in the lab. He's now left the lab. He's an assistant professor at Harvard Chemistry now. But Brian came in the lab and he looked at these cells and he became curious about RTKs. So glioblastomas have, 2/3 of glioblastomas have an alteration in an RTK, amplifications of PDGFRA or EGFR. Yet the inhibitors have entirely failed in the clinic. And this is something that Brian was puzzled by. And he thought, can we use some of these models to address this? So Brian actually went and studied a whole number of these glioma sphere, glioma stem cell lines that we have in the lab. These have been sort of generated through collaborations with neurosurgery and Hiro Wakimoto at MGH. But we now, and also other colleagues in the field, we now have quite a panel of different glioma sphere lines from different patients that we expand in serum-free media, kind of the same media that you would use to grow neural stem cells. Brian asked what happens if you treat these with a PDGFRA inhibitor, dasatinib. And this is just sort of the viability curves. And as you might expect, we have lines that are EGFR amplified. They don't care about this drug, right? They're perfectly fine. We have lines that are MYC amplified. They don't care about these drugs. We have a bunch of lines that are PDGFRA amplified. To be transparent, some of them don't care about this drug. But at least some of them, and here's an example of this GSC8 line, some of them are addicted to PDGFRA. And when you hit them with dasatinib or another PDGFRA inhibitor, they crash, just as you would have predicted, right? Yet remember, the therapies aren't working in the clinic, so what's going on here? Well, the answer is that most of the cells crash, but you leave behind about 8% of the cells in a persistent population that are able to remain viable even when you give them a lot of dasatinib or a lot of crenolinib or whatever drug you're using to PDGFR, or even if you knock down PDGFRA, a small group persists, okay? And if you let these persist for a while, eventually they start growing out a very slow cycling population of glioma spheres that are drug tolerant. They don't need, they're PDGFRA independent now, even though they were originally addicted. And we're calling these persisters sort of in light of work, initially from the Settleman lab with lung cell lines. But here we're looking at glioma spheres, glioma stem cells. Now, one striking result is if you now remove the dasatinib, these slow cycling persister GSCs will, within a week, switch back to the proliferative state. They'll regain their dependency on PDGFRA and they'll start growing very rapidly, okay? So in a sense, from this one cell line, and we've now done this with several other lines and spheres, we can isolate two different states that don't seem to be genetically different. We think they're isogenic, and I could give you some evidence for that, but that they're epigenetically switching between alternate states. So let me tell you a little bit about these states. First is that these persister slow-cycling GSCs upregulate histone demethylases. So histone, I talked to you a little bit about these different marks, but one of the memory marks that go onto chromatin to help maintain their states is methylation of the histones. And so there are classes of enzymes that put on those marks, and there's classes of enzymes that erase those marks. And it's sort of been suggested, and there is evidence from model organisms, that the enzymes that remove those marks are like epigenetic erasers. They kind of remove memory. And we were struck by the upregulation in these, and this has been seen in certain cases with stress. In this case, maybe it's the drug stress. And in particular, we were interested in the demethylase family that removes this repressive H3K27 mark, methylation mark. Remember, this is the one I talked a little bit earlier about EZH2, this repressor that we think a lot about in cancer. EZH2 puts on K27 methylation. KDM6 removes it, right? So EZH2 tries to shut off genes. KDM6 lets them come back on again. It erases that repressive mark. So we used small molecule inhibitor of KDM6, and we used various CRISPRs. We actually used the VACOC a method where we actually took CRISPRs and we directly targeted the catalytic domains of KDM6. And what we found is that the naive sort of proliferative GSCs they didn't need KDM6 at all. It's expressed at low levels, but we could knock it out and the cells will proliferate just fine. But these persister cells are highly dependent on KDM6, and they are very sensitive either to the inhibitor or to the CRISPR inactivation. Okay? Good. Why do you need to remove K27 methylation marks? I could spend a lot of time on this, but I won't. I just want to tell you an example of a locus. This is HEY1. And HEY1 is silenced in those highly proliferative cells. But in the persister cells, what happens is it's silenced. The gene is off. There's no active signal. And then here is this repressive signal. There are these peaks of repressive chromatin right over the gene and over a nearby enhancer. And those peaks go away when you switch into the persister state. So we think that the demethylase is going around sort of removing repressive chromatin and allowing certain things to get reactivating. So what's getting reactivated? There's a couple things getting reactivated. A lot of primitive master regulators. SAL2 is coming way on, and a lot of genes you think about have very primitive neural stem cells. And the other thing that's getting reactivated is Notch. The Notch pathway is clearly coming on, and you see lots of Notch targets, both Notch target enhancers and Notch target genes are coming on. We think, in part because we're now erasing the chromatin and facilitating this switch. As you might expect, with Notch coming on, the persisters are very sensitive to GSI inhibitors of Notch or to genetic suppression of Notch by dominant negative mammal, whereas the naive cells, they don't have Notch, they don't care about GSIs, they're impervious to any of these things. You might ask, how do these cells emulate what's going on in the tumor? And this is very important to us. We spend a lot of time characterizing tumors. We characterize tumors at single cell levels by RNA-seq. We characterize tumors by IHC and IF. And so I want to just show you that real quickly here. So there clearly are, in human GBMs, there are persister-like cells. So, and I'm sorry that this didn't map up perfectly, but this is sort of a multispectral imaging showing different colors for different cells in a primary GBM. And the key point here is that there are cells that are Ki67 positive in pink. These are sort of proliferative cells in a tumor. And there are orange cells that are staining for Notch activity. This is the intracellular Notch, ICN1. You can clearly see staining for ICN1, and the marks don't seem to overlap. So it seems like these Notch state suggests a very slow cycling state. The tumors are relatively slow cycling anyway, but the Ki67 proliferatives, they're non-overlapping. But the key result, I think, here is that you have these Notch positive cells within the tumor that are clearly tumor cells. And we also did single-cell RNA-seq, and we did a lot of analysis where we now are using gene signatures to see what looks like what. And the only thing I want to point out here is that there's gene signatures for naive GSCs and persister GSCs, and with each column here is a different cell from a tumor. And what's clear is within tumors, we can find a group of cells that have signatures of persisters. They upregulate KDMs. They have turned off cell cycle. And you can see a signature of Notch signaling in these cells. So again, at the RNA-seq single cell level, there are clearly cells within primary GBMs that emulate and look a lot like these persisters. Okay, so just to wrap this up a little bit, what I'm telling you is that it looks like glioma cells, particularly glioma stem-like cells, can exist in a proliferative state that's RTK dependent, but they can interchange with a persister state that's very slow cycling and seems to be Notch dependent. I'd like to quickly point out that in normal development, embryonic development of your brain, there are neural stem cells that are quiescent and Notch dependent, and they interchange with transient amplifying cells that make up the parenchyma of your brain, and they are RTK dependent. So in a sense, I might suggest that your glioma tumors here are borrowing a page from normal development and able to now reversibly transition between these states, perhaps with the help of KDMs and these erasers and the plasticity with which they're endowed. I think this conclusion is probably pretty obvious to this group, but you might imagine that chemotherapy and targeted therapy may get this subset of glioma stem cells and other cells in the tumor. But I think you'd agree that these persister cells that are very slow cycling are going to be very hard to get by conventional modalities, and we need to think about alternate strategies, either with KDM inhibitors, which are not yet, at least this one's not yet really at the drug stage, or perhaps by GSI Notch inhibitors. The idea is that epigenetic plasticity is contributing to tumorigenesis at different stages, right? So in tumor initiation, right, you're starting out with some kind of neural stem cell. We can debate what the cell is, but it's probably a slow cycling neural stem cell in your brain. There's these barriers that chromatin enforces, and it keeps that stem cell in its appropriate state. But if you get a lesion, and I exemplify IDH mutation, but I would love to have the conversation. I would argue that there probably are also non-genetic things that cause to screw up your chromatin environment and can lead to plasticity. But in this case, the most illustrative, rigorous analysis relied on an IDH mutation. This IDH mutation reduced the barriers here and allowed this neural stem cell to start, adopting or sampling different cellular states, turning on different genes because you're losing different insulators. In most cases, the states are inconsequential. It didn't matter. But at some point, some neural stem cell is going to switch over into this proliferative state with activation of PDGFRA. And it's going to become proliferative, right? And it's going to expand the population. And through acquisition of a couple additional mutations, it's going to lead to a tumor cell, right? But maybe there's something more, right? Now you have a cancer cell and it's proliferative. But, here it's proliferating. But I would sort of argue that, maybe the tumor microenvironment stresses. We know they upregulate KDMs. So now you're going to even more erase the epigenetic memory, right? You're going to induce more plasticity. So this proliferative tumor cell may be at a sample even more further alternative states. And this state here, I think, is particularly interesting. I would posit maybe it's a de-differentiating further a little bit more into something that looks like a quiescent neural stem cell that's very dormant, and you treat this patient with chemotherapy and whatever you have, you get this initiating cell, you get all these other cells, but this guy has drifted into this alternate, very primitive refractory state, and you're missing it. So two ways in which plasticity is contributing to tumorigenicity. We should, I think, pay attention here. That's it. I just want to thank colleagues in the lab. Will Flavahan and Yotam Dreyer led the work on the IDH mutations and the loop and loss of oncogene insulation. Jem Sievers, Brian Lau, and Anu Patel led the work on drug resistance in glioblastoma. Glad to take questions.
Thank you very much. You stayed well on time, so maybe we have a few moments for a few questions.
The experiment where you did the CRISPR mutation of the CTCF binding site near PDGFR, could you say a little bit more about what you learned about its effect on multiple genes in that area and what you were able to learn about how that impacts either histone methylation or other epigenetic marks when you take away those constraints of that loop in that area?
That's interesting. We have not really done that experiment yet to look at all the other global alterations. There are several genes in the locus, and you can imagine there may be other disruptions. I should point out that Rick Young's group had another locus that had a CTCF, and in their model, they think about genetics. And in their model, they show that a genetic mutation to a CTCF turned on a developmental gene in a TALL. I don't think either of us have rigorously looked at whether the refolding of the chromatin alters the regulatory landscape and how it does. We've really focused on how it alters the three-dimensional interactions that might be allowed or prohibited.
I think your model of clonostochastic selection after the epigenetic lesion is really nice and has to occur, and it's a really nice way of looking at it. But I was just wondering a little bit about the complexity. If the clonal selection is based on activating the PDGFR, I still think you'd get inhibition by IDH inhibition. But there can be lots of other types of clonal selection. Like you're activating a lot of genes with the initial hit, some of which may be suppressors and other things that decrease things. So you can get loss of those, or you can get other lesions that are genetic or epigenetic that enhance the PDGFR. And in those, you'll be resistant you'll have a real hit and run. And I wonder if you can divide, you see this in patients, right? Some are IDH sensitive, inhibitor sensitive, and some aren't. And I wonder if you and others must be looking globally at what happens when you get the, the selection process, which can take years.
So I want to be very clear, and I may not have made the case strong enough, and maybe I'm wrong, but this is what we kind of think. We think by the time you've switched this insulator over to a methylated state and you've lost CTCF, that is a stable event. And it's in IDH, remember you kind of suppress the TETs?
Right.
I think allowing the TETs to go again, they don't have any effect. In fact, we went in the lab and we used IDH inhibitors hoping that we could reset that loop. They did not touch the switch. That was methylated, CTCF was gone, and we couldn't reverse it. When we used 5-azacitidine, it quickly reversed.
Right.
So based on our work in the lab, it is a hit and run. By the time you've made that switch, you don't need IDH anymore. That's the first thing. The second thing is, might there be other events that contribute to PDGFRA and other things? Absolutely, I totally agree with that. The third piece is, I think just in glioma, and obviously in leukemia, IDH inhibitors are incredibly exciting. I have not heard much about IDH inhibitors being useful in glioma.
I totally agree with that part.
Okay, good.
I just wonder about the leukemia part.
So incidentally, we can use TCGA data to start asking which tumors seem to have insulator disruption driving them, and we hit a couple tumors. We did not hit leukemia. It may be that that's an entirely different mechanism. It might be entirely different leukemia. So that's why I was very careful. I said, look, we've got this in glioma. It looks like it's something in GIST is similar, and we might have one other tumor now, but I have no idea what's going on in leukemia. Yeah.
Hi. Do you perceive this as a mechanism for all the copy number alterations? Not necessarily all, but the massive number of copy number alterations that are coming out from the TCGA, the CTCF methylation.
Well, let me think. So I think when you have a copy number application, that's separate. That's just genetic. What I'm arguing is that in the absence of copy number alteration, you could turn out that locus in a purely epigenetic way. So I think now you're asking, we have about 50 oncogenes. Correct me if I'm wrong. Maybe there's 100, maybe there's 20. But there's probably 50 oncogenes that get that are amplified and turned on with a gain of function. I just threw that number out, but anybody have a better number? Say there's 50 oncogenes that we know genetically are driving. I think an interesting question is how many of those could be activated by loss of a loop insulator, by purely an epigenetic mechanism?
I think it's a very interesting mechanism, but you would think also if you've got that activation, that's going to lead to more mistakes that could lead to gene amplification. It would seem to me.
Well, that's true too. So there's. This is why the field's so complicated.
CRISPR experiment
right.
and look and see if over time you acquire gene amplification.
I've tried to simplify the presentation, but there's no question that what really causes cancer is not just genetic, it's not just epigenetic, it's genetic and epigenetic. And what you've learned is that genetic lesions can increase epigenetic plasticity. I could also make a pretty good argument that epigenetic lesions can make a cell hypermutatable or can lead to genomic instability. So, I think we need to try to, there's a lot more we need to study. And, it may well be the case. We don't know yet.